Our next presenter is Dr. Deborah Golino. Deborah is director of the Foundation Plant Services since 1994 here at UC Davis. Deborah's also a cooperative extension specialist in the Department of Plant Pathology at UC Davis, and she is active in teaching and extension and service in addition to administrative duties here at Davis. Her research program is directed at controlling the virus and virus-like diseases of grapevine within a focus to detect and streamline virus elimination techniques for clean stock and quarantine programs. And she's an author of over 100 scientific papers. And Deborah, we appreciate you being with us today.
Deborah, I think you're muted.
Can you hear me now?
Yes, you're good.
Okay, good. Great. Well, I want to thank everybody for including me in this webinar. We certainly have had a lot of good talks, and it was very interesting to hear that we may have a confirmed vector. I'm excited about that, actually. So I'll be talking today about FPS and the National Foundation Plant Services and the National Clean Plant Network, protecting the supply chains of grapevines from red blotch and other viruses. There we go. I'm the director of Foundation Plant Services, which is a self-supporting department at UC Davis, and we have historically, back into the 1990s, held the the largest national collection of grapevines available for commercial nurseries to make planting stock. We have a number of responsibilities. We're here to produce, test, maintain, and distribute elite disease plant propagation material. We provide plant importation and quarantine services, virus testing and elimination, and then we coordinate the release of UC patented horticultural varieties. And then we do a lot of outreach work linking researchers, nurseries, and producers. We have quite a few crops here, but the most visible is our grape work. So what's the basic idea of a clean plant program? New grape selections come in to our program, and they could be foreign imports, they could be domestic selections, or they could be new varieties like the rootstock varieties being developed by Dr. Andy Walker. We take those new grape selections, we disease test them using some of the technology you've heard about today and other tests as well. If virus is found in plant material, and our program is all about virus and virus testing, we do disease elimination therapy. So this is a program that helps control viruses in planting stock, because viruses in planting stock can easily go into plants that are propagated from virus-infected material at a very high rate. So after disease elimination therapy, disease testing occurs again. If all the tests are negative, then the individual session goes into the program. It's planted in the foundation as a provisional vine. Professional identification is done to make sure that the cultivar or variety is correctly identified. And then those registered foundation vines go to nurseries and growers. So when red blotch hit, we were very concerned, as you might imagine, that our materials in our foundation vineyard might be affected, and we did have evidence that there was red blotch at both nurseries and in growers' fields. How do we do the pathogen elimination? It's an important part of our discussion of the new Russell Ranch Vineyard Protocol 2010 and the National Clean Plant Network. What we do when we do pathogen elimination here at FPS is we take a shoot tip. Let me get my cursor going here. We take a shoot tip from a growing cane of an actively growing grapevine. We cut it off, we surface sterilize it, and then we, under a microscope in sterile conditions, we cut a meristematic piece that's 0.5 millimeters or less, and then we regenerate a new plant. That can take six months to a year, depending on the cultivar grade. When we do that, it gets rid of almost all of the grapevine viruses. That's a part of a very long process. You see here, this is the simplified version of the grape importation process here at UC Davis. What I want to do briefly is have you take a note of how many years this process can take. It's a multi-year generational process to create a collection of virus-screened grapevines. We bring them in, we do the tests as I've said, we do identification. If something comes to us perfectly clean, it can be 5 years before a nursery has registered material. So it's not a quick process, and steady funding and consistent staff are very, very important to success. Now, in the 2008 Farm Bill, there was money for what is known as the National Clean Plant Network, and that is funding to help specialty crops create the best possible foundations of virus-screened material for their industries. And the National Clean Plant Network includes funding for cherries, for grapevines, for citrus, for hops, for berries, and this year we've even added roses and sweet potatoes. So it's very important funding to do this long-term screening of important plant material to make sure that we improve our the profitability and sustainability of our specialty crops here in the United States. And the notion is to create this virus-free material so that it is readily obtained by nurseries across the country, who therefore will make better plant material for our commercial growers. With that funding, in 2010, we established a new Russell Ranch Foundation vineyard where every vine in the collection had been through micro-shoot tip culture. What was the idea here? The idea was that if you perform micro-shoot tip culture, you're getting rid of viruses that you know about, and you're also getting rid of viruses that you don't know about. And it was because of the National Clean Plant Network money we were able to do that. We established the new vineyard in 2010. By October 2012, you can see some of the vines of Scion Wood are growing quite nicely. And even though red blotch had not been described until 2012, we had the solution in hand when it was the news first broke about red blotch, because we had this new Russell Ranch vineyard started with USDA money from the National Clean Plant Network. So the concerns and frustration of industry with the questions that we can't answer yet about red blotch are very understandable, but we were very, very lucky, and in fact, in some ways, I think we've dodged some bullets by having established this Russell Ranch Foundation vineyard. Everything in the Russell Ranch Foundation vineyard is at the Protocol 2010 level, which means it's all gone through micro-shoot tip culture, and it's all been negative for a long list of virus tests, which is actually growing every year. And we go back and retest vines in the Russell Ranch to make sure when a new virus disease like red blotch is added to the testing routine that the material is free of the virus. So what about red blotch? You can imagine my concern as director when the news came out about red blotch, the fact that we were seeing it in both nursery and grower fields, that what about our collections at Davis? Do we have red blotch in our collections? And I want to pay tribute to Dr. Deborah Golino, who is in charge of our labs, and his wonderful staff, who very quickly started turning around incredible numbers of virus tests for red blotch so that we could get the answer to that question. Now, our classic foundation vineyard, the vineyard that existed before the Russell Ranch vineyard, the new one, the classic foundation vineyard is very large, it's still very active, and at the time we started testing was still very important because the vines in Russell Ranch were still way too small to provide material to nurseries to put into their new increase blocks. We tested in a short amount of time 4,361 vines in the classic foundation vineyard, and of all those vines, only 15 were positive for red blotch disease. That's a super low number of vines to be positive for a virus that we didn't think we were screening for. And I have an explanation for that I'll share with you in just a few minutes. In the meantime, what did the positive vines look like, and which ones were they? This is a list of the vines that were infected with the virus. There's no clear pattern here about what was infected and what was not. It included both red and white grapes. But what was super interesting to us is when we looked at the pattern of where these vines were, they were randomly scattered all over the vineyard. But for each positive vine, there was one vine of the same clone that was negative for the virus. And that suggests that this is a very unusual virus in many ways. And I believe that Keith Perry referred to this too. Red blotch just doesn't quite behave like all of the other viruses we've worked with for over the years in grapes. And so we've still got a lot of work to do with it. Now, the symptoms were relatively mild in the foundation vineyard. We do regular inspections looking for symptoms of virus in our foundation, and I think that perhaps we probably already eliminated by visual inspection maybe a few other cases. But you can see here on the left is a healthy Chardonnay. On the right is a red blotch, is a red blotch infected Chardonnay. And then down below, Ruby Cabernet with and without the red blotch virus. This is the infected on the right. Not very strong symptoms. Now, these are own-rooted vines, and I'm guessing we'll find, as we have time to do more research on red blotch, that we will find that own-rooted vines show milder symptoms of red blotch than vines on their own roots. Vines on their own roots will show milder symptoms of red blotch than vines that are grafted onto rootstock. Now, didn't we have more virus in the classic foundation? Here's what I think. I think the woody index, the field index that we've performed at FPS since the 1950s was picking up red blotch as well as the leaf roll viruses. And a good example of that would be the Zinfandel selection that went to Switzerland. It was sent to Switzerland as a leaf roll positive sample for a virus collection in the 1980s, before we even had laboratory tests for the leaf roll viruses. The reason it was sent is it was positive on the woody index for Cabernet Franc. So let's talk about the index a little bit, and then I'll show you some interesting data that I think proves my hypothesis. What you do for a woody index is chip bud 2 buds of the candidate, the vine you're trying to test, into a minimum of 6 indicator plants per variety. And the indicator plants are St. George rootstock, Cabernet Franc, and LN33. Then you plant those out in the field and you observe leaf and trunk symptoms. And I'll go through that quickly. It's a very visual thing, but I do want you to understand the process. We disbud, wax, root, and pot up indicator plants. We chip bud 2 buds per plant, 6 plants per variety. We cut buds off dormant canes of the candidate variety. We cut a matching notch in the indicator plant. You can see it right here. Place the candidate chip into the notch, wrap with budding rubber bands, and as you can tell, this is very labor-intensive work, and my tribute to the staff that do this here on our horticulturalists are really good. You harden off the plant, put it out in the field, read the bud tape to see if you actually got the candidate to live on the indicator, and then you go out in the field at year 2 and observe for symptoms of leaf roll or red blotch. And this is a picture of me and Larry Bettiga, our viticulture advisor from Salinas, and we were looking at the variation in symptoms of leaf roll and red blotch in Cabernet Franc. There's quite a bit of variation, actually, virus strain to virus strain. And that's something I want to caution people. We shouldn't generalize about what red blotch virus does. We have to think about the fact there's likely to be symptom variation and differences in effect of different strains of red blotch, just like there are with different strains of red blotch, of leaf roll. So here's leaf roll in the field in a Cabernet Franc, And up close, you can see the green veins that Sudhi talked about. The year after the announcement was made about the discovery of red blotch, we went out to the field, our lab did, and did PCR tests for leaf roll and for grapevine red blotch virus on all of our positive Cabernet Franc vines. So things that would have historically been diagnosed as leaf roll, we went out and we tested them for both viruses in the lab. So that there's the positive for the Cabernet Franc index, and you can see here leaf roll PCRs. We always thought there were some leaf roll diseases that we hadn't found yet, because we always had, and you can see right here, a group of negative for leaf roll PCR plants that were positive on the Cabernet Franc index. We found that year that every single time we had a positive plant that was negative on the leaf roll PCR, it was also positive for red blotch disease. And also, interestingly enough, just because a vine was positive for red blotch didn't mean it wasn't positive for leaf roll too. And of course, on the Cabernet Franc index, you'd just see red leaves. You wouldn't know whether you were seeing red blotch or leaf roll. But I believe that field index is what really saved us, and that's the reason that we don't have more red blotch in the foundation collection than we do. So what about red blotch in Russell Ranch Foundation Vineyard? Russell Ranch Foundation Vineyard is 100% red blotch free. We also tested every vine there, and we are now routinely testing vines that are planted there. So that's all good news. One question that really interested me and my colleagues is red blotch a new disease? Dr. Maher Al-Rahani helped me with this project. We had him go over to the herbarium on campus, the UC Davis Center for Plant Diversity Herbarium, which has over 300,000 dead plants, leaves and stems, whatever is the most reasonable sample to take, sometimes flowers, which are flattened and dried and glued onto archival paper with a label. It's a technology that botanists have been using for a long time. The label on each one has a specimen, cultivar, place, date, and collector's name. It even has some great samples that are unmounted. Here you can see them on the old newspapers in which they were wrapped. So a fully documented accession has details like the ones you see here, specimen number, determination, collector, etc. And Dr. Harold Omo, who was a really wonderful scientist here at UC Davis that was a grape breeder and plant explorer and did many other things for our grape industry in the United States around the world, he brought in a lot of the grape selections that are in the herbarium. Now, the scientists at the herbarium were willing to let us take 46 grapevine specimens, older specimens, and to take a little bit of tissue off of them and to collect that to take back to the lab to run for red blotch virus. We made sure that only fresh materials were used. We didn't want any contamination. It's too easy to get cross-contamination with the red blotch virus. 56 sample bags were transported to a lab in which research on grapevine had never been conducted. We used all fresh reagents, and this is a list of some of the samples. All new materials and reagents for every sample, and one sample of that group of 56 was positive for grapevine red blotch virus. It was an early Burgundy sample collected by Harold Omo, and the year of collection was 1940. We also obtained the full genomic sequence of the herbarium isolate, and we compared it to recently sequenced red blotch isolates. All of the work was done in a medical lab, which had never worked with grapes also. And the full genomic sequence of this herbarium isolate shared 92 to 99% nucleotide identity with the other gene bank isolates to which we compared it. I've got that paragraph twice, and the results suggest that red blotch virus was present in the Sonoma wine grape growing area for at least 74 years before the virus was identified and correlated to specific symptoms. So red blotch has probably been with us a long time. It's very interesting to hear that it does not seem to be in Europe and that the vector may be a North American insect. Fascinating, in fact. And with that, I'd like to thank the team of scientists and staff at Foundation Plant Services who have labored very hard to help turn the red blotch story around as quickly as possible. The American Vineyard Foundation, the IAB, the Nursery Advisory Board, and the California Grapevine Nurseries, all of whom help us fund this work at FPS. Thank you.
Thank you, Deborah. I have to say, you really that was an interesting story, and it was wonderful the way that FPS responded to the issue with red blotch so immediately. It was a good example of how that should work.